Okay, so we should all see a little red light in the upper left-hand corner, which lets you know that your session is being recorded. And with that little bit of housekeeping, let me turn the presentation over to the NovoBio 3D printer team.
Hello everyone, we are group 3. Today we'll introduce our NovoBio 3D bioprinting solution. The outline of our presentation will be, first we will outline our Hedgehog concept. Then we'll provide you with a product overview. Next, we will describe to you how each subsystem addresses a set of corresponding customer needs. Then we will highlight what makes our bioprinter unique. Next, we will provide a summary of our financial analysis. Last, we will explain to you why our design is worthy of prototyping in Mechanical Design 3.
So our Hedgehog method is to pursue a design that's compatible with a diverse array of cell types safely, and the features that allow this are pointed to with the orange arrows on your screen. We also aim to produce a bioprinter that's adaptable through its simplicity as requirements may evolve in the future. The three pillars of the Hedgehog concept are excellence, driving of one's economic engine, and passion. And the main motivation behind this Hedgehog concept is really our passion to maximize the device's potential to aid in life-saving research. Very simply put, the more people who can use this and the more cell types that it's compatible with, the more potential it has to be a part of saving lives down the road. To address this Hedgehog concept, our design incorporates a quick switch printing tip, a highly adjustable bio-ink pressure while printing using a stepper motor and syringe pump system. And it's also designed to accommodate a massive array of volumetric flow rates down to a very small 5 nanoliters per minute. These points of fine adjustability allow for accurate printing of even very small and frail cells.
In this section, we will overview our product. Our product consists of 7 distinct subsystems: the dispensing mechanism, the printing head motion stages, the motion adjustment mechanism, the support structure, the print head adjustment mechanism, the external pump system, and finally, the remote power transmission system.
In subsystem 1, dispensing subsystem, a customized printing head is designed which can be installed onto the motion platform through attachment. The threaded external surface of the dispensing tip and the internal surface of the printing head attachment structure will be connected by simply screwed in. The dispensing tip will be made from clear resin by stereolithography printers, and the printing needle dimensions such as length and diameter can be customized based on customer's desirable flow rate and ink viscosity. Switching printing head may take less than 10 seconds, therefore providing installation and replacement convenience.
Our design uses 3 linear stages with dowel pin bearings to control the XYZ motion of the dispensing mechanism. The motion is driven by an 80 TPI adjustment screw with a flat machined onto it, as shown in the red circle in the middle of the presentation, to provide mechanical engagement with a set screw for power transmission along the shaft. A right angle bracket is used to assemble the stages and XYZ configuration to provide up to 12.7 millimeters of travel in each axis. The angular deviation of each stage is expected to be less than 150 microradians, which performs far beyond the customer's needs of less than 1 degree.
Slide, we will be discussing the support structure and attachment mechanism. The support structure subsystem consists of 2 parts: the turret mount and the L bracket frame. The turret mount will be press fit into the turret fixture, and the L bracket will be fastened to the motion stage. Both parts are made of 6061 aluminum. The L bracket is an OTS part that can be purchased from a MasterCard. The turret mount can be manufactured easily. The two parts will be attached by two screws.
There's also a resin printed plate which attaches to the final linear stage via a couple of screws. And this serves as the attachment mechanism to attach the dispensing head to the motion platforms. Initially, this was going to be a separate structure to that plate, but we combined it to reduce the number of parts. The print head simply threads into the plate using a lure lock style mechanism, which securely holds it in place. One of the big advantages of this solution is that it eliminates the need for fasteners, which could introduce metallic wear debris to the print job.
The external pump mechanism pumps fluid to the print head by means of a linear actuated syringe pump. The stepper motor drives the shaft threads, which engage with the external threads on the syringe. And this moves the plunger up and down to pull in fluid and push it out. The fine thread spec of the pump and the stepping abilities of the motor and control board allow for accurate flow control and flow rates to meet the stringent customer requirements. The pump is manually loaded by the operator with a quick snap-in-place holder to prevent it from rotating during usage.
Our remote power transmission consists of 3 flexible steel motor shafts that connect the linear stages to the 3 STP MTR 23055 stepper motors. The stepper motors provide 17.25 pound-inch of maximum holding torque with an inductance of 2.4 millihenries, thus making it compatible with the smoothie board provided. Both ends of the flexible shaft are female fittings designed with set screws for secure attachment. Both the stepper motor shaft and the linear stage shafts have a D slot to promote mechanical engagement with the set screws. The motor is mounted to the motor mount with a mounting bracket and fastened down to the base. In addition, the stepper motors are capable of performing micro stepping, which will allow for greater precision in translation and controlling the torque. In the second section, we will overview how our device was designed to meet a rigorous set of customer needs and how much it cost us to do so.
There were a total of 7 subsystems used to address the customer's needs as shown in the flowchart. Key performance features of our design include flow rate as low as 5 nanoliters per minute, linear speed as low as 0.1 micrometers per second, and achievable feature size resolution of 100 micrometers. This flow rate, along with the linear speed, may be dynamically controlled to print a wide variety of features and feature sizes within the limitations of the 96 well plate printing medium. Attractive features for customer include low manufacturing and assembly cost of just over $1,000, low system weight at 104.75 grams, and Smoothie Board 5X compatibility for use of in-house software development at UF.
The total cost for our design was calculated separately for each subsystem. The most expensive process is buying the OTS parts, which accounts for slightly over half of the total cost. The second most expensive process is the manufacturing cost, which accounts for about 35% of the total cost. Because most of the materials are purchased off the shelf, this explains the low cost in our raw material costs. And also because we prioritize using snap-in-place parts and minimize the use of fasteners, our assembly labor cost is also very low. Overall, our total cost adds up to $1,022, which is about a quarter of the $4,000 sales price. This really maximizes our profit. Brings us to our final section, Lastly, we provide you with a synopsis of the many unique features our design has and why these unique features mean it should be selected for prototyping.
Our design has three major features that are unique from other current designs: an elliptical syringe pump, user-friendly print tip, and flexible motor shafts. Due to the geometry of the syringe pump, a guide rail that you would typically see on most linear actuators is not required. This really eases manufacturing, and it also prevents shear of sensitive biomaterial. Additionally, it snaps easily into place without tools. We optimize flow control by use of a smoothie control board in conjunction with fine driving threads. The printing tip is user-friendly due to its ease of replacement and install, reducing effort required by laboratory technicians and therefore minimizing mistakes that they might make during that process. The transparency of the tip also allows the operator to confirm correct flow calibration by making bubbles in the system visible. Lastly, the flexible motor shaft gives the operator leeway during setup so that there's less wear and maintenance required for the system's operation.
So why our design? I'd like to reiterate 4 key reasons why you should select this design for prototyping. As discussed previously, it has a lot of innovative features. It has an incredibly low production cost of just over $1,000. And this is significantly lower than the $4,000 we were allotted. It's very light, coming in with all microscope mounted parts at just above 100 grams. This is also significantly lower than we were asked to do. And all components are designed really with the user in mind. Basically, our design takes the customer needs and not only meets them, but exceeds them. And it does that while costing a fraction of what we were asked to do. So thank you all very much for your time and for your attention. At this point, we'd like to open the floor for questions.
Okay, thanks for sharing your presentation with us. Do we have any questions from the gallery?
Hi guys, this is Elise from Cummins. I like how compact and relatively minimal number of components there are in your design. And I thought you did a good job with presenting that. I just had a question. Did you do any analysis on your like L bracket or your press fit or anything else like that?
Yes, so the L bracket, so we calculated the deflection at as you can see, the maximum deflection should be towards at the end of the L bracket, like in the Y direction. And we calculated that the maximum deflection is less than 1 cell diameter, which means that the accuracy is not really affected by the deflection. Therefore, like the L bracket, it's like a good choice for us.
Thanks. And so is the press fit is where the, what is it, extrusion tip is, or is that, where is that again?
Well, it's the dispensing tip.
Okay, yeah. Did you calculate the stresses in that at all?
Actually,
So
like a lure lock style thing, which threads into place rather than just a straight press fit.
Okay, so it's threaded, not press fit.
Yeah, yeah. As you can see on that picture on the left of the dispensing tip, those ridges there, those are threads.
Wait, so which piece was there a piece that was press fit or?
Initially, we were going to press fit this, yes. And that's actually a design change that we made recently. We find that this lure lock style threaded mechanism is a bit easier to deal with for a lab technician and also a bit more secure holding.
She might also be referring to the snap in place mount for the syringe pump, if you want to go to that picture. So it's a little hard to see because it's not zoomed in. If we had CAD, this would be a lot easier. Where it says effortless snap in place syringe mount, it's not a press fit, but it requires deformation of the part to snap it into place, which is ultimately what holds that into place, but it not necessarily a press fit.
Did those answer your question, Elise?
Yeah, thanks.
Cool, thank you.
So I've got a quick question on the design, guys. So it looked like for your Z axis, you had the cable coming right down at the bottom of the Z axis. Yep, right down there. So do you have anything planned for how you're going to keep that from actually interacting and interfering with whatever's on top of the microscope? Because it looks like that's coming down and actually projecting right alongside almost where your print head is.
Oh, it's due to the view angle. The printing head will be aligned with the optical axis of the microscope, and it can be, since the dispensing tip length can be adjusted by the customer, so it will be reach the well plate, and the part which controls the linear motion stages with a connector at the bottom. Since it's flexible, it will be bending to other directions, and in order to not cause interference with the microscope working place, the length of the printing tip will be enlarged in order to reach the well plate based on the real situation of the microscope.
To add on to what Chen said, that
With
currently for the
the
FDR, we're producing some guidelines about where we can locate the stepper motors in order to maintain function of the flexible shafts, because there's an allowable bend radius that they can be before they stop rotating about their central axis, specified by the manufacturer. And we're also going to consider those sorts of things, not interfering with the microscope plate, etc.
all right, cool, thank you.
Yeah, I have a question.
Go ahead, Daniel.
Do you have a view of the lure lock system that she was talking about? It looks like it seats up pretty far into the, go back one slide, I think. I don't know, you passed it with the syringe? Okay, there we go. So it looks like it seats up a decent amount into it. I just didn't know how much room you have to grab onto it to twist it in there or to thread it in. So I didn't know if there was, if that was problematic, or I know that the gasket was going to give you some depth to seat into, but you know, you got to be able to twist it. So I didn't know if that was something that.
Yes. So the hand will, if you are able to see my mouse on the screen,
Yes.
the hand will be holding the end where the needle is connected with the head, and that part will be not completely submerged or screwed into the support because the seal will be elastomer between the printing tip with the holder, and therefore there will be a space for hand to be manually handling and inserted.
Okay, and then this is that, I guess that's whenever you tighten it down, then it's going to compress some certain amount or whatever, and the it's obviously going to squish and then reduce the diameter of that hole. So I didn't know if that was an issue either, or if you have plenty of ways to ensure the flow rate it stays where you want it to stay, or is
Yes.
that above the hole? Oh, I see.
So
Okay, never mind. No, I apologize. It sits above the hole. So yeah, you can seat it up into it. All right, thank you.
I may have missed this, but do you guys have a picture of the entire system? It seems like there were, and then where do those tubes go to? Do you have that as well?
so this tube
Yeah.
is going to the syringe pump here,
Okay.
and these flexible shafts are going to be connected with the motor, the remote power transmission subsystem.
Okay. And how do you guys attach those? Are they just on the table or are they in a box?
They are installed on a motor mount, and the motor mount can be located on a table. There are plenty of flexibilities in laboratory use because the shafts are flexible. They are soft.
Right, but is the plan to have those items as loose items, or are you gonna pre-mount them in some sort of container?
We're leaving
Oh,
that up to the customer. But we are gonna, like I said, I mentioned before, we are gonna produce guidelines, in the process of doing that right now, of producing guidelines of where they should be located for optimal performance. But we thought that we considered having some standard place to put them, but keeping them open like this allows for a lot more customizability and ease of use within different lab settings depending on their configurations.
And how are these powered?
these motors are wired onto the smoothie board and using the controller.
The smoothie board uses a 24-volt power supply.
So by each of those motors are then separately powered.
The
Have
motors
separate
are all
cords?
the 4 motors are all connected onto the smoothie board.
I have one more question on that slide. So how do you know where the positioning is? Because is there a feedback mechanism or something to tell you how far you've pushed or pulled or anything with the stepper? Or like encoder or anything, or is that not important in this subsystem?
So this current remote power transmission, we have the calculation on how far the motion state travel with each rotation the motor generates.
Yeah.
And therefore, the software will be controlling the motor. The software can be run through any G-code software that control 3D printers or CNC machines. It's the same mechanism.
Yeah, no, I totally get that. I was just wondering, like, position wise, when you turn it off and turn it back on, if there's any type of homing or something that goes on so that you know the position relative.
That's
So that you can tell
controller
me how
side,
many.
Daniel.
What's that?
That's handled on the controller.
Oh, cool. Okay,
Yeah,
thank
the
you.
smoothie board uses end stops, which basically when you see it's used pretty widely on 3D printers. When you see each axis
Current
move
spike.
to its limit, it's zeroing itself.
Gotcha. That's sufficient. Thank you.
Yeah, sure.
So I assume the smoothie board isn't shown here.
Yeah, the smoothie board is what we have in the lab that we're providing as the controller for all these devices. So we told them it has to function and interact with the smoothie board, but the smoothie board was not something that was a part that they were required to design. It was something they were
Okay.
required to design to be compatible with.
Gotcha. Okay. Thank you.
Okay. Well, we've got time for maybe 2 more questions. So if anyone in the gallery wants to ask, now is the time. Okay. Well, seeing no questions then, we will wrap things there. So let me stop the recording process.